Yeah.
All right. So no, I can't see my notes, but I don't have so many notes, so I think I can do without.
Okay. Wait 1 minute and then we'll get going. Just to keep on track exactly.
Yeah.
At least according to my clock.
So I can say thanks for inviting me. That doesn't have to be a part of the formal talk, but thanks anyway.
So yeah, I'll just introduce you. So Geir Halnes from Norwegian University of Life Sciences is our final speaker of the session on ion concentration dynamics in brain tissues. So Geir, the floor is yours.
Thank you. So it's kind of a broad title, I suppose. So it's effects on neurodynamics and extracellular potentials, and the two are kind of different when it comes to modeling formalisms needed. And I'm in this group of a guy called Geir Halnes. And the main expertise in this group is on modeling extracellular potentials. And so I'm going to start a bit just putting up the standard framework used for that. So when you want to model it, you typically model the neuron using some kind of multi-compartment model, for example, based on the neuron simulator. And then you compute the neurodynamics using that. You get currents going in and out of the membrane. And then when you're done simulating the neurons, you can, in step 2, feed these transmembrane currents into the surrounding tissue as just, you forget about the neurons and now you just have a set of distributed sources and sinks. And if you have such a distribution of sources and sinks, you can actually find an analytical solution for the extracellular potential that you will have outside this neural activity. And this equation is based essentially on current conservation, so that you will have closed loops between all these sources and sinks. And just to compare it to the previous talks that have been given, I think all of them were essentially single compartment neurons, and which means that you only have an inside and an outside, and you have no space inside the neuron. And in that case, you would not have any extracellular potential, because you would have to have, because of current conservation, you will have to have the same current going in to the neuron in form, for example, of a sodium current, as going out from the same point in the form of a capacitive current charging up the membrane. So you would have a zero sum, not zero voltage dynamics, but zero current, and then no extracellular potential. So the extracellular potential depends on the distribution, so that in this case, you can have current coming into the soma, going up the axon or dendrite, and out somewhere else. But you do this using a two-step scheme. And the main assumption in such a two-step scheme is first that there's no feedback from whatever the neurons causes the extracellular potential to do, doesn't act back on the neurons, because you have simulated the neurons in an independent step. And normally you assume this is okay, because the extracellular potential is so small compared to the membrane potential that it wouldn't affect much anyway. That's the assumption. And the second assumption is that ion concentrations are constant, as you have talked about today, and which is also the main topic of my talk. And a third assumption is that this extracellular current is ohmic. It's given by Ohm's law. And this is a prerequisite for deriving this equation that you see down here. And based on the previous talks, I also think that you know this fairly well, but I'll repeat it quickly. So the reason why we sometimes can assume constant concentration is that if you look at the concentrations inside the neurons, they pretty much sum up to a zero net charge. So the number of anions and cations are more or less the same. And so there's a small deviance here, 0.2, Maybe that's an overestimate, but it still is like on a decimal place. And this net charge inside the neuron, it's actually not floating around in the bulk solution because that's electroneutral. It's sitting on the membrane, and it gives rise to the membrane potential, which for resting neuron is something like minus 60 millivolts. So what happens during an action potential is that some sodium rushes in. It changes, for example, the sodium concentration by 0.3 millimolars. And then it repolarizes, and then some potassium goes out and changes the potassium concentration, reduces it inside. And the assumption is that between this action potential and the next, the pumps have time to pump back these reset the original concentrations. And also the assumption is that these tiny concentration changes during an action potential are so small that you don't really have to bother with them. They don't really change anything in terms of neural dynamics. And all this, of course, fails in some cases where you have hyperactive neurons that are firing too fast for the pumps to keep up, or if the pumps in themselves are not working, such as after a heart attack or a stroke or decapitation. So when are these concentration effects relevant? Of course, they are very relevant in very tiny spatial scales, such as inside spines, ion channels, synaptic clefts, and very close to membranes and the bilayers. And this is not my focus. I focused on the larger spatial scale, and that's when neurons fire fast over time, and you get an accumulation, for example, of potassium in the extracellular space. Potassium seems to be the most critical, as I agree with the previous talk on this. And then you get changes in reversal potentials. And also, if you really want to do this spatially, you get changes also in currents going through extracellular space. They are not ohmic anymore, they are also diffusive, meaning that there will be currents proportional to concentration gradients because ions are charged, and when charges move, it will be an electric current. And so part of my title was extracellular potentials, and I've devoted only this one slide to it, and I focus later more on the neural dynamics aspect. So we've focused in quite, well, 3 papers on how diffusion can affect extracellular potentials. And we've kind of simulated, and I'm not going to present the formalism behind this, but we simulated it in this way. We took a complex neural model, but standard in terms of implementation. It's a neuron simulation in a model from Hay and co-workers. And we ran it for 80 seconds simulated time, and we computed the currents going over the membrane at all different neural compartments. And we split it into sodium, potassium, calcium, and unspecified and capacitive currents, and we kept track of all the ions going out into this box of extracellular space. And then we looked at the concentration due to electrodiffusion in this extracellular space. You see this red blob surrounding the neuron. This is the change in potassium after 80 seconds of simulation. And this panel below shows for a specific location during an action potential, the extracellular potential in one case where we accounted for the diffusive effect on it, and in one case where we didn't. So this orange line would be the standard theory, and the blue would be if you account for diffusion. And what you see is that they're essentially parallel, but diffusion has only shifted it by like a small amount to a more negative value in this case. And so this shift, you could picture that in the beginning of the simulation, where potassium was more or less constant in the entire space, these would be on top of each other. And then during 80 seconds of activity, changing the potassium concentration, the concentration has shifted the potential by this much. So it's a very slow effect. But if you look at the power spectra for this with and without diffusion, you can actually see that for high powers, the two cases are on top of each other, but for lower power, you see they split apart, so that you could expect diffusion in some cases to show up in your power spectrum if you're measuring local field potentials and things like that. So that was my first part of the talk. That was, I called it an introduction, and I put all this extracellular stuff in the introduction. And now I go on to a kind of general mathematical framework for dealing with these kind of things. So you can think of this problem as having three different kinds of currents: those that go across the membrane, those that go inside neurons, and those that go outside. And the membrane currents, you know well, Hodgkin-Huxley type, and the only ghost of ion concentration is baked into this reversal potential, and you normally assume it's constant in standard models. And intracellular, you have the cable equation, voltage gradient, the currents follow that, and the same in the extracellular. The difference is that you have 3D. So what happens when concentrations start changing is that, as you have seen in the previous talk, reversal potentials can change, and that will change firing properties. And so you get this depolarization block and stuff like that you can simulate. And if instead you get gradients in the extracellular space, I focus on the outside here, you cannot use Ohm's law anymore to study it. You have to use the Nernst-Planck equation for electrodiffusion, where you have a flux of ions going because of the concentration gradient, CK, and another one because of the potential gradient, because if there's a potential gradient, it will propel ions along with it. And that should replace Ohm's law from the standard models. And just to look at the relationship, I can sum over all fluxes weighted with their valences to get a diffusive current and a field-driven current. And then I can compare this to Ohm's law, and it should be the same if this diffusive component is 0, and if this prefactor, concentration-dependent prefactor, is equal to sigma, the conductivity, and if that's constant. And so when we want to solve this, we have to solve this equation. And if we say, maybe ideally, we would like to just demand that this should be fulfilled everywhere in space. And we have two options, because this will give you one equation for each ion species, but we need one additional equation for this additional variable phi, which is the voltage. And we have two options. One is to use Poisson's equation, which derives phi from the charge density locally. And the problem with this is that it's extremely computationally efficient, because this charge density accumulates on a nanosecond, nanometer scale, and you have to have an extremely fine resolution. And there's an easier way, and that is to just assume that all solutions are electroneutral, and so that the charge density should sum up to 0 everywhere. And to explain, I'll jump to the next slide. To explain the difference between the two, I can try to explain what the diffusion potential is. So if you picture that you have a box with two ionic solutions of different concentration, consisting of anions and cations of the same amount on each side, and you put them in contact with each other at t equals 0, time 0, what you will expect is both to start diffusing towards the right, towards the lower concentration. But if you look up in tables, you will see that, for instance, if this is sodium chloride, you will see that chloride has a larger diffusion constant than sodium, so that due to diffusion, more chloride will move than sodium. And this is a quite dramatic thing, because now you have a net charge transfer from one side to the other, and it will give you a charge difference between the right and left box. And this charge difference will set up a potential, almost a step potential between the two sides. And this step potential will slow down the chloride ions and speed up the sodium because they're oppositely charged, so that you get a 1-to-1. And this you will get after just about 10 nanoseconds or so. So that difference between these two frameworks is that the Poisson-Nernst-Planck, you actually have to simulate this very fine-grained charge relaxation process, while in this electroneutral scheme, you just calculate the step potential needed for it to become 1-to-1. So it's a shortcut, and it can be shown to be accurate on timescales larger than 10 nanoseconds, and at least on the micro millisecond timescales that you are interested in when simulating large-scale neurodynamics. So the essence of this framework is essentially just to solve the Nernst-Planck equation in the intra and extracellular space, but you don't want to solve it in the membrane, because that's like a very complex place with the bilayers and everything. So instead you couple them on the membrane using Hodgkin-Huxley type boundary conditions. And you can compute the potential by demanding these two, by putting in these two constraints, that for any compartment not bordering to a membrane, there should be no net charge at all times. And for any compartment bordering a membrane, the net charge should be exclusively associated as a membrane charge, and the membrane charge will give you a membrane potential. So there should be a strict relationship between the charge in the in and outside and the membrane potential. And so maybe that's, from a technical point of view, that's the model we are most proud of now, because we have a way to do this for a general 3D geometry. But the problem is so you can see here we have meshed up both the extracellular and intracellular space, and we simulate ion concentration dynamics in both compartments and make sure that the membrane currents are consistent with that. And the problem with it is that it's extremely computationally expensive. So if we want to run simulations on it, we are limited to just very small idealized geometries so far, and maybe brief simulation, maybe some hundreds of milliseconds, and a few pieces of axons or something like that. And so in a sense, I think it's a bit boring to play around with models like this, because you have to start to simulate. We have to do a lot of work to set up your simulation, and you have to trust very much the parameter choices that you made, and then you have to wait for a couple of days, and then it spits out a result. So it's more fun to do a smaller model where you can just run a lot of simulations and play around and see what happens. So I'm going to focus this last short part of my talk on just commenting on a minimal model that has it all. So we wanted to see if we can make a full multi-compartment model which is completely electrodiffusive in its nature, meaning that it accounts for ion concentration dynamics, spatial propagation, and has a consistent relationship between all ion concentrations and voltages everywhere. And by minimal, I mean that multi means two in this case, and that we have different ion channels in the soma and dendrites, so it has some spatial structure and can get somatodendritic signaling. And also we have a spatial explicit extracellular space, but also here only two compartments. And that this model should account for all ionic movements due to electric drift, diffusion, and transmembrane currents. And so the way that we proceeded trying to make this model was to take an existing two-compartment model. So we used this Pinsky-Rinzel model, which is fairly well established. It has two compartments, a soma and a dendrite, some potassium channels and calcium channels in the dendrite, the standard action potential channels in the soma. And then we asked, can we make this ion concentration, can we combine this with ion concentration dynamics in a way that makes it provide the same functionality in terms of firing pattern as the original one, which means that we cannot assume that ion concentrations will remain constant, but we actually have to make sure that they do. And I wanted to comment on something before I go on, and that when we equipped this with an intra and extracellular space, we assume that the extracellular space has half the volume of the intracellular, which is a tissue average. Neurons occupy about 40% of brain tissue, glial cell 40%, and the extracellular space 20%, so that extracellular is small. And when we do this with closed boundary, as I also think that to some degree other single compartment models have done, we actually assume periodic boundary conditions, meaning that when my neuron pumps out some potassium and it ends up and stays in this compartment, why doesn't it diffuse away from here and use the whole space? Well, in this sense, it wouldn't if the neighbor was doing exactly the same, because then you would get the same concentrations everywhere. So this is, it should be noted that that will represent the scenario where you have perfect synchrony in a population. It was just a comment. Apart from that, I think this model is nice, but this gives you this particular interpretation of what it should mean. And so just some few take-home messages from this model. What I found interesting was first to see how long time it actually took to get a resting state. As you maybe know, if you have made these standard models, normally if you don't know exactly what the resting potential in your complex model will be, but you just run your simulation and let it settle on some resting potential. If you're lucky, it has one. And that normally in a normal model, it takes a few hundred milliseconds perhaps. So you let it just run for 100 milliseconds before you start doing something with it. But in this case, when you have ion concentrations that can go back and forth and even inside and outside, you have a lot of different variables that need to find their collaborative resting state, and these concentrations are very slow. So what I found in this model, what we found was that we have to run it for more than 1,000 seconds of simulated time before it actually gets entirely to rest. And it doesn't even change much during these 1,000. It changes by a few millimolar and finds its own resting state. It has a resting state, and just to show, or like one criterion for this to be an acceptable model was that it should reproduce the spike shapes from the original model, and it did. It's not very surprising since it contained the same ion channels, but so now we have a more complicated model that does exactly the same, which is normally nothing that you applaud. But at least in this case, you can compute a set of new variables that you couldn't in the previous model, like so concentration shifts during spiking and conductivity shifts in the extracellular and intracellular space, energy consumption due to how much the pump did pump, and the extracellular potential. But the main point was that, and this was something that we hoped would happen and expected to happen if the model was sound, that if we stimulate this with a weak input so that it's firing slowly, it changes during firing the potassium concentration, but it settles on some dynamic steady state, and it can go on forever spiking, and it can spike identically to the original Pinsky-Rinzel model. But if we stimulate it harder, the concentrations will drift off gradually, so it will spike identically to the original model in the beginning of the simulation, but after a while the concentration changes started to show up in the spiking patterns, and in the end it reaches the polarization block. And I think I saw something like this in David and Lausanne's talk too. And since I am running out of time, I'm jumping over this. I wanted to just make my last comment on the tendency in the field of decoupling electrical drift and diffusion, because standard models of electrodynamics normally you only consider drift currents, which means only the ones that are driven by the potential gradient. While if you want to model concentration dynamics, you often do the opposite. You only consider concentrations changing due to diffusion. So, and we asked during, or the motivation that for looking into this was that I spoke to a colleague who wanted to study some sodium dependent stuff going on in dendrites, and he wanted to model diffusion of sodium from soma towards dendrites. And I wondered, is that a meaningful thing to do? Because there are all these electric currents going in that direction, and they are also carried by ions that are there, and maybe sodium moves just as much due to drift as diffusion. So we asked, during this particular simulation that you see here, what causes ions to move from soma to dendrite, drift or diffusion? And the result is shown here. Blue is drift and orange is diffusion, and the panels show a cumulative movement of charge or ions. And I think this colleague was right. In the case of sodium, it was mainly diffusion that drove sodium upwards. But in the case of potassium and chloride, which have higher concentrations on the inside, by the way, you can see that at least drift and diffusion are of comparable magnitude, and so on. So I cannot conclude that these two phenomena can generally be decoupled as you would like to do. We have expanded this by adding a glial cell outside, and I'm not going to talk about that. It's available in a preprint. And thank you.
Thanks a lot, Geir.
Sorry for going over time.
We do have time for questions, and we have one in the chat. So this one comes from Mark V. What are the exact conditions for using KNP versus PNP? It must depend on the length scale as well, presumably.
Yes, that's like the troublesome question. But I think if you use PNP, you need to model the actual accumulation of charge in a Debye layer. So typically, if you do that around a membrane, that will be like 10 nanometers or something like that at max. So you have a concentration gradient. So you need a spatial resolution of smaller than nanometers and a temporal resolution smaller than nanoseconds. And if you don't use that, your system you will get all these instabilities, ringing and stuff like that. So it's very impractical, but it's very accurate. KNP, you can just choose any sort of temporal and spatial scale. It will not be as accurate, but it will be stable however you choose it. I hope that was a somewhat satisfactory answer. KNP, you cannot study phenomena taking place just immediately outside membranes and inside ion channels and at a very fine spatial resolution. But like on a tissue average or whole cell level, I think it will be fine.
Okay. Another question is this 1,000 second requirement for reaching steady state is quite striking. That's quite a long time. Is this because your standards for being converged are really, really strict, or is there can you comment on any biological effect that requires this much time?
Do you still see my screen?
Yes.
Okay, so I'll just, I've lost it myself. Okay, so it's not really strict, but anyway, you can see the convergence here somehow after the concentrations have changed due to a period of spiking. So you can see they start leveling off towards rest. And in this case, the time scale is 6, so it's a minute. So in this case, it takes about a minute to reach. And I think our criterion for convergence was something like reached delta concentration should be less than 1% or something like that from something you get after 5,000 seconds.
Okay, so it sounds like this 1,000 seconds is not an absolute number, but it's more a function of what type of transient and stimulation precede the converging.
Indeed, yes. But in general, I mean, the point was just that it's like on a minute time scale, I think you could say, where you can expect concentration effects to linger in your system for a minute before. So it has a memory. Concentrations gives this cell a memory of a minute, in a way.
Okay. We've reached the end of our time. I don't know if there's any other quick question. Okay, yeah, well, it is noon here, or 6 p.m. I guess in continental Europe. So that reaches the end of the session. So thanks very much to